couple disclosures I have. I am involved in many different studies. I do have a leadership role in a couple of the upcoming studies, including one that's going on now, the Roche Generation 2 HD study with Tom and Nerson. I'm on the steering committee for the Novartis PTC study that we hope will be coming up in the US. I'll talk about that. We get some funding from the Griffin Foundation. I'm a scientific advisor for Azavan, doing research in aggression in HD. I'm a scientific advisor for Teva, and we get funding from CHDI Foundation for the Enroll study, and I'm also a scientific advisor to them. So I'm going to talk a little bit about our center. I will note some of the accomplishments since we were founded back in 2013. I'll talk a little bit about the HD biology so you understand what's going on with some of these trials. And then I'll finish up just featuring a couple of the clinical trials that are either ongoing or in planning process right now. So we heard a lot today about HD-CERC, which is what we call our center. So it stands, of course, for care, and I hope we've shown you how comprehensive our care services are. Education, we do a lot of training, certainly a lot of research, and we've met our research team. And again, we are a center, so we are housed at Georgetown. We do a lot of our work at Georgetown, but we also do outreach. So we understand that coming to Georgetown may not be fun. It may not be fun to battle the traffic and park at Georgetown. So we do have other locations where you can see us where it's much easier to get in. So in terms of that, we have the MedStar Montgomery Center out in Olney, and we have the center at McLean, Virginia. And both of them are lovely because you can drive right up, you can park next to the building. The Olney Center is probably the most handicap accessible place I've ever worked in my life because you can literally see the office from the parking lot. So it really makes it a much better experience. You can do the Enroll study, the observational studies at these sites. For any of the drug studies, the interventional ones, you do have to go to Georgetown. But to establish clinic care, if you have a relative who hates the idea of going into Georgetown, come out to one of our satellite clinics. So who are we? This is Dr. Fahad Amjad. He's a movement disorder neurologist, meaning he has specialized training in Parkinson's, Huntington's, and all these other conditions. He's our clinical co-director. This is Dr. Steve Lowe, and he's out at the McLean, Virginia office, also a neurologist with movement disorder training. And Dr. Fernando Pagan, who's the head of the Movement Disorders Division at Georgetown. This is Emily Weaver, who you met, our social worker, who's literally the glue that holds everything together. Robin Kaprevich, who's on camera duty, well, was on camera duty today, who's our trials director. This is Dr. Blancato, whom you heard this morning, again, our geneticist, who also does genetic counseling. This is Lisa Ebcheski. You met her this morning in our physical therapy talk, and she's in clinic seeing patients for consultation for free. And this is, of course, Joey, who's planned our fabulous education day. This is Erin Koppel-Caruso, who's our clinical trials manager. And this is our senior research assistant, Mara McCartan, who's been manning our table today, and myself as the center director. So again, we have a big role in educating clinicians. For those of you who come to the clinic, we often have like little students trailing along like little ducks behind us. But it's fantastic because for most of them, it's the only chance they get to meet somebody with Huntington's and hear about the disease. So later on in their career, if they take care of someone with Huntington's, they have some idea of what the disease involves. We do a lot of didactics, which are lectures. So we lecture to residents, we lecture to medical students, and we do lectures for other professionals who aren't specialists. Emily and Joey do a huge amount of outreach to long-term care facilities, educating them about HD, educating their staff on how to take care of people with HD. And then as you heard today, we do a lot of student research so that they can learn about research methods and how to do a study on their own. So in the last years since we were founded in 2013, we've done over 20 interventional studies at our center. These are drug studies where people have the chance to try a treatment. And then we've had these observational studies also. We've had 4 of those where there's no treatment, but we're collecting information. So we've really been able to bring a huge contribution to the DC area. As someone mentioned earlier, there was no opportunity to do any kind of HD research or care before our center was founded at Georgetown. So just a little bit of background on HD biology. I know Dr. Blancato talked about this morning. So you have chromosome 4, and there's this expansion that happens that causes these repeats, right? And we think of, 36 to 39 as the intermediate area, 40, definite HD. And we have this normally folded protein. So if you have less than 35 repeats, your Huntington's protein is normal. If you have more, you get a misfolded protein. And that causes at least some of the pathology in Huntington's disease. So that's why it's important to know about this. So what's the function of this Huntington protein that we talk about all the time? We actually don't know, right? So we have this protein that's expressed throughout the body. We all have Huntington protein, so it's not something that you only have if you have Huntington's. Everybody has Huntington protein all over their body. It's essential for development. So if you take a baby mouse and you do a genetic thing to it so it doesn't have any Huntington protein, it will actually die before it's born. So you need Huntington protein to develop. It does something really important, we think, with brain development. As I said, it's found all over the body, but the highest concentrations are in the brain, which is probably why the brain is so affected in Huntington's. So it has all these different things where it may be involved in signaling between brain cells. It may be involved in bringing proteins together to do what they need to do. It may be involved in protecting the cell from dying too early. So brain cells are programmed to die at a certain point, which is a normal thing. But in Huntington's, we know that they die before they should. Maybe it plays a role in repairing damaged DNA. So all kinds of things like radiation, sunlight can damage your DNA. Maybe Huntington protein actually helps repair that. There have been a couple of studies showing that people in Huntington's families have a lower rate of cancer. So it may be that it's actually doing some really good things for us. So I know there's a lot on this slide, but I'll take you through it. So you have this Huntington protein. It actually gets cleaved into this thing called exon 1 and fragments. And that is what causes some of the problems also. So you get these aggregates, meaning clumps, that are in the nucleus of the cell. And then you get these other clumps that are out in the cytoplasm of the cell. And we think that it causes all of these different problems. So we think that, again, you get dysregulation of protein transcription. There's a thing called DNA mismatch repair, where, again, the DNA needs to be repaired if it gets broken for some reason. Maybe it gets repaired the wrong way, and you get expansions. Then you have this thing called somatic expansion, which means in certain tissues in the body, especially the brain, your CAG repeats can take off and really expand. So if I test you and you have a 41 repeat in your blood, if I come back and test you in 20 years, you'll probably still have a 41 in your blood. But if I biopsy your brain in 20 years, you may have like 120 or 200 in your brain. So we think that these big expansions of CAGs that happen in the brain and some other tissues are also part of the reason that we get all these problems in HD. I'll read the rest of that. So now we have some disease treatments that I'm going to talk about. So we have what are called antisense oligonucleotides. We have splice modulators and RNA interference, and I'll show you where they fit into all of this. So the first part I'm going to talk about are Huntington silencing therapies. So these are drugs that basically turn off Huntington's protein, right? So they just turn off the switch so that you're not making Huntington's protein. So these, again, are called antisense oligonucleotide. That's the first category I'm going to talk about. So these drugs basically stick onto the messenger RNA, and they can shut down production of Huntington protein. So the idea is that you get this drug at some point in your life, it shuts down your Huntington's protein, it sort of gives your brain and the rest of your body a break from the Huntington's protein, and then compared to someone who didn't get treated, the red line, who's not doing as well, you do much better, so your symptoms don't progress as fast. That's how they're supposed to work. We're not at that point yet, but that's the idea behind these therapies. And I've got my little picture up there with the person to show that these are given through spinal taps. So a lot of these studies are going to involve spinal taps. So the first bunch of ASO studies that were really studied in humans in large scale were the Roche tominersen studies, also called Generation HD1 and now Generation HD2. So we had this study from Roche. It was a huge study around the world looking at this way to lower Huntington protein, again, just shutting down production of all Huntington protein, both normal and abnormal. So we all have normal Huntington protein. People with HD also have abnormal Huntington protein or mutant Huntington protein. And this drug was knocking down all of it. Unfortunately, in March 2021, when we were all in deep depression from the pandemic, Roche decided that they had to close the study because it seemed that the drug was actually making people worse for certain patients. So it was very discouraging, but it was obviously the right thing to do because some of the people on treatment seemed to be doing worse than people who were not getting treated. So Roche, to their credit, looked at the data more, and they found that people who were younger and not as advanced in disease might actually have been getting some benefit from the drug. So this idea that people with more disease, maybe their brains are not as resilient, they can't handle the drug, but maybe the ones who are younger and don't have as much disease can tolerate the drug and actually get some benefit. So that's what led us to this new study, Generation 2 HD. It's in a younger population of people who don't have as much disease. They're earlier in the disease. So that if there are some side effects of the drug, perhaps their brains can tolerate it better. So that study is ongoing right now. We're hoping to get results in the next year or so to see if that will actually help people. So I talked about how we all have Huntington protein. Again, all of us in this room have Huntington protein. If you have Huntington's disease, you have both normal and abnormal Huntington protein. There's a company called Wave that has figured out a way to lower only the abnormal protein and leave the normal Huntington protein around to do what it's supposed to do. So here is wild type, which is what they call normal Huntington protein. So in someone who's healthy, you've got wild type protein and you've got stress, and they sort of balance out. In Huntington's disease, you've got this decrease in wild type because you have some of the abnormal protein, right? So your brain sort of has this balance between normal and abnormal protein. Unfortunately, when you put stress and mutant Huntington protein together, it's a bad combination, and that's what leads to the dysfunction in the brain and the early cell death that I talked about, and the shrinking in the brain. So the idea is that you want to get rid of the bad Huntington protein, but preserve the normal Huntington protein because it's doing something good. So Wave has this complicated process where they take a blood sample and they can basically, they have this little marker called a SNP, and the SNP, if it's located on the same gene or allele as your Huntington's expansion, it serves as kind of a little marker, kind of like an Apple AirTag saying this is where it is. And that makes it so they can only lower your abnormal Huntington protein. So they have done some preliminary work. They had Wave 1 and 2, which did not work. So it was specific, this drug that could latch onto this little Apple AirTag that did not lower mutant Huntington protein. But they now have a study that seems to show about 46% lowering of mutant Huntington protein. And we're waiting to see if they will start opening sites for that. The one question I have is that there is some evidence of NFL, or neurofilament light chain increase. That's a compound that we think of as a sign for cell distress or cell damage. So there's some question about whether there's some toxicity with the Wave study. So that's why we're waiting to see about that. Again, Spinal Tap up there showing you how you get this drug. Then there's a company called Unicure. So they've taken a virus and they've made it so that it's not a bad virus, it's not giving you a cold or an infection or anything, but they use the virus as like a little messenger, like a little car to get their drug all over in the brain. So they do brain surgery. They make holes in your brain, kind of like deep brain stimulation surgery that we talked about before. They make holes in your skull, they put a thin needle into your brain, and they inject their drug, tagged onto this virus. The virus goes all over the brain and distributes their drug, and the drug lowers Huntington protein, both normal and abnormal. It also targets that exon 1 fragment that I mentioned that may be involved in these big expansions that we see in the brain. So on the one hand, it's brain surgery, but it's only one time. So you're not coming in and getting a spinal tap every 4 to 6 months to get your drug. On the other hand, if there's a negative effect, it will last forever. So that's the balance of this. If it works, it will be long-lasting. If it doesn't work, if there are bad effects, it will be long-lasting. They do have a small number of subjects who've gone through this. They have some really promising clinical results, so we're waiting to see where they go with that. The last study I'm going to talk about is the PTC study. So this one, you notice, has a pill jar. So this is the one study that you can take by mouth. You don't have to take it through a spinal tap. It's a small drug, and what it does is it basically tells, it goes in and it tells the pre-mRNA to stop. So it's like it's reading along, it's reading a story, and then it puts a stop sign in the middle of the story instead of at the end. And that stops it from making Huntington protein. This one will lower both normal and abnormal Huntington protein. It's developed by a company called PTC along with Novartis. So there was a company, so Novartis previously had a compound called branaplam, which was somewhat similar. They were doing a study of this in Europe, and they had a lot of side effects, so it was stopped. People were developing irreversible neuropathy, so they were losing fingers and they're feeling their fingers and toes, which is bad. So they stopped that study. Fortunately, this new compound from PTC is different enough that it's not causing those side effects so far. Some of the preliminary results they have, the safety's been really good. They've been lowering Huntington protein by about 40%, and they seem to actually be slowing the production of NFL, which is that distress signal that neurons send out when they're sick. So that's been really promising. So I talked just very briefly now about ASOs, about splice modulators, RNA interference, which was a uniQure study. And of course, there are other things that are either here or coming down the line. So there's the sigma-1 agonist, which is from the company Prolentia, which seems to have a neuroprotective effect on the brain. There is this idea about the DNA mismatch repair. So there are a couple companies looking at FAN1 and MSH3 targets for this mismatch repair. And that may also lower the somatic expansion, the big CAG repeats in the brain. And then, of course, we always wonder about CRISPR and gene editing, right? You just go in and fix the gene and not worry about all this downstream stuff. And I know there's a lot of work in animals. To my knowledge, there's nothing that's quite ready for humans yet there. So these are the different mechanisms that we at least know about, or at least someone's working on right now. So in summary, I hope you've seen today how our center integrates research and care. We have a multidisciplinary team. We hope you take advantage of seeing many or all of us, multiple locations, so hopefully one of them will work for you or your loved ones. And there's a lot going on in HD research and a lot more to come. So I just mentioned a couple studies that are in the clinic right now. We have many, many more companies that are interested. When I was at the CHDI therapeutics meeting earlier this year, there were over 60 biotech companies that were there, and they're all in different stages of development for HD. So it's a really promising time in HD. So in the end, I'd like to thank all of you, especially you families, who took your time out from your lives to come here today. I'd certainly like to thank our team. It's a fantastic team. I couldn't do any of this without the team. And the Griffin Foundation for all of their wonderful funding and support over the years. Thank you.